Okay. Hello, my name is Paulo de Castro Reis, and I'm the Executive Director of CCBC, the Chamber of Commerce, Brasil-Canadá, based in São Paulo, Brazil, and with an office in Montreal, Canada as well. I'm pleased to open this event today, and I would like to thank all attendees and speakers for being here. We at CCBC are always committed in strengthening the relationship between Brazil and Canada by bringing perspectives from both countries in the most relevant topics across the multiple sectors. Specifically in the health sector, CCBC has an active committee involving Brazilian and Canadian companies and experts. If some of you participating today at this event are interested, please feel free to contact our CCBC team. The threat of COVID-19 and the new variants is in everybody's mind. And this event will address the current issues in this area and bring best practices developed by both countries. With that, I would like to call Ricardo Meirelles, who will be moderating this event today. Hello, Ricardo, welcome.
Hello, Paulo. Thank you so much, Paulo, for the intro. Yes, I'm very happy to be here to discuss such an important topic and also had the opportunity to talk to such top professionals in the healthcare space that will be sharing their time with us today. Before I introduce our first speaker, I want to say thank you for some people who really helped us to put this together and work behind the scenes. Some names to acknowledge here: Ademar Seabra, Trade Commissioner; Dr. George Khalil from University of São Paulo; Dr. Daniel Garros from University of Alberta; Stefan Ganet, CEO of Ovenza; Fernando Sembranelli, CEO of Health Innova Hub; Marcelo Greco, CEO of the Green Hub. And from the FIOCRUZ team, who we have today here, Dr. Claude Pernes. I also want to thank you, Dr. Tania Araújo, Dr. Marco Aurelio Krieger, Dr. Milton Moraes, who have an active participation in this initiative, and Cleide Garces. And with that, I would like to introduce our first and our keynote speaker today, Dr. Eric Marcusson, co-founder and chief scientist officer of Providence Therapeutics, first Canadian organization that developed the COVID-19 vaccine with the mRNA platform. And with no further ado, I would like to pass to Dr. Erickson.
All right, thank you very much, Ricardo, and thanks for inviting me to speak here today. And so I want to give a background on mRNA vaccines specifically for COVID. I'll only be touching on the mRNA vaccines, and I think that some of the other panel members will talk about some of the other mechanisms of vaccines. So we'll first start with just an overview of mRNA vaccines for COVID, really a little bit about mRNA vaccines in general, and then the two that are approved for COVID. And then I'll just touch briefly on the development of our own vaccine, PTX COVID-19B. So what is an mRNA vaccine? This shows you the components. Obviously, an mRNA vaccine has to have an mRNA in it. And so that's the part of the vaccine that encodes the antigen that leads to the immune response. And it's made in a chemical, an enzymatic process called an in vitro transcription reaction from a DNA template. So to start the whole process, you first make a circular piece of DNA, and you use that as a template to make your mRNA. And down below is the structural characteristics of an mRNA. So the 5' cap, 5' UTR, 3' UTR, which UTR stands for untranslated region, and the poly A tail are all structural parts of an mRNA. And so these UTRs and poly A tails, you can use the same one for different antigens that you encode. And then there's what's called the ORF, or open reading frame. And this is what encodes the actual antigen. In the case of COVID-19, all of the mRNA vaccines are against the S or spike protein of SARS-CoV-2. And each company's ORF is different, even if they have the same antigen, because of the way that mRNA encodes proteins. There are what's called codons, and a 3-base codon codes for an amino acid. And each amino acid has multiple potential codons that can be used. And all of the companies, instead of using the nucleic acid sequence of SARS-CoV-2, try to improve upon it. And they do what's called codon optimization. So if you looked at Moderna's vaccine and BioNTech's vaccine versus our vaccine, even though we're all encoding the S protein, we're going to have different nucleic acid sequence or mRNA sequences in there. We'll also have different 5' UTRs and 3' UTRs. All of us will have a cap in this case and a poly-A tail. And the poly-A tail is usually about 100 A residues long. And then what's important then is that it has to go through multiple rounds of purification for the mRNA. Obviously, you want a very pure drug or vaccine. And so you go through multiple rounds of purification and then encapsulate it in a 4-component lipid nanoparticle. And that lipid nanoparticle is very standard these days for the general scheme. What's different between the different vaccines is that ionizable lipid. So each mRNA vaccine company is going to have their own ionizable lipid. They'll all have cholesterol, the same cholesterol that's in your body, and then a helper lipid. And there's very few helper lipids, so most companies have very similar or identical helper lipids. And then what's called a PEG lipid or polyethylene glycol. And the PEG is what's shown here in the schematic. And this is important for the stabilization of the LNP and also the distribution of the LNP. And then inside that LNP, or basically it's a fat bubble. It has cholesterols and lipids, the same like lipid nanoparticles lipid high density lipoproteins and LDLs in your body. It's a fat droplet, and inside that fat droplet is where the mRNA is stored or contained. And this lipid nanoparticle protects the mRNA and helps it to get inside the cell, because the mRNA is a very large molecule and it won't cross inside the cell where it needs to be to get translated into an antigen without this lipid nanoparticle. So the lipid nanoparticles are a very important part of the mRNA vaccine. And everybody's lipid nanoparticle is slightly different in terms of their construction, but all of us have lipid nanoparticles if we're doing mRNA vaccines. You really can't do it without it. And so what are the advantages of mRNA vaccine platform? Well, the mRNA technology has emerged and developed really quickly. I mean, it's been around for a long time, but COVID has accelerated the development, right? There were no approved mRNA vaccines before COVID, but mRNA vaccines had been worked on for quite a while, and they were moving through the clinical trial process, but there wasn't this great urgency that caused them to move so fast. And the mRNA vaccine is the fastest way to get a vaccine. That's what we saw with the first 2 vaccines that were approved for COVID. The reason why is once you have the genetic sequence of the virus and you know which antigen you want to go after, as I mentioned, the spike protein for SARS-CoV-2, it's very quick to design the mRNA. I mean, everybody has their own algorithm, and you just put in the protein sequence and it spits out the mRNA design. And you just have to use that to make the DNA template that I talked about, and then make the mRNA from the DNA, and then formulate it and finish it into vials and ship it out. And that whole process can be done in 8 weeks. So from the time you know the sequence of the organism to the time you can be testing in humans, you can do that in 8 weeks. And Moderna proved that in COVID, where they started their trials about 8 weeks after the sequence was identified. And what's really nice also about it is the ability to dose many times. Unlike a viral vector vaccine, like for COVID, AstraZeneca and J&J have viral vector vaccines. Those can be dosed twice at most, and by the time you try to dose a 3rd time, the immune system of the subject will see that virus right away and neutralize it before it has a chance to do what it needs to do. And so that's a problem with, we're all talking about the variants of concern and the possible need of boosters. It's going to be very difficult for a viral vaccine to do that. It would need a new virus. Basically, you have to have a new viral vector if you want to dose a third time. And scaling and manufacturing of mRNA is relatively simple compared to the other technologies. And you can see how quickly it was able to speed through clinical development. I mean, once you're actually in the clinic, there's not a big advantage to the mRNA vaccines, but getting to the clinic is what gets accelerated. So the mRNA vaccines that are out there for COVID-19, there are two. I think most people know them by now. They're extremely well used throughout the world. So there's the Pfizer BioNTech or BNT162b2. There was the first vaccine that was gained emergency use authorization in the United States, and then spread from there throughout. Actually, I think it was first approved in the UK, I believe. It was later approved, just like a week later, approved in the US, and within weeks throughout many countries in the world. It was found to be about 95% effective at preventing symptomatic COVID-19 disease. It's given as 2 shots, 3 weeks apart. And as I said, it's now available, in many countries around the world. It appears to be effective against the variants of concern, including P1, which is the variant predominant in Brazil. It does have a reduced immune response to these variants, but the immune response, the original virus, is so strong that even after reducing the response a little bit to these variants, it's still very likely to be neutralizing. And there's data coming out now about that and showing that although it's not 95% effective, it's probably somewhere in the 80s. You need a little more data to be sure about it. The Pfizer vaccine, one of its weaknesses is that it's stable for only a short periods at 4 degrees centigrade. So it does make it a little harder to get to rural areas. It needs to be frozen for all but except I think it's about 3 days that it can stay at 4 degrees. And so it doesn't give a lot of time to get shots in the arm once it's thawed. The second vaccine to be approved was the Mrna Vaccine, also known as mRNA 1273. 1273 is the number of amino acids in the S protein, so that's where it got its name. And it's basically equally effective to the Pfizer BioNTech vaccine. It's also 2 shots, but it's given 4 weeks apart. And it's a little bit of a difference in 4 and 3, but it's really a difference in what the companies chose. Either one should work fine for an mRNA vaccine. I don't think there's a right answer whether it should be 3 weeks or 4 weeks. We do ours 4 weeks like Moderna does, but 3 weeks is also very effective. Moderna is also available in many countries around the world, not quite as broadly distributed as the Pfizer vaccine, but pretty close. They're making billions of doses of this vaccine now and trying to get it spread throughout the world. And just like the Pfizer vaccine, it appears to be effective against the variants of concern. And the one nice thing about the Mrna vaccine is it's stable for a longer time at 4 degrees. So you can have a little more time to get it into arms, but it does seem to have a slightly more negative tolerability profile. Both vaccines are definitely tolerable and certainly they're safe, but you do hear about people having some reactions and fevers and chills and headaches, and it's definitely a side effect, but that's what happens when you get your immune system activated real strongly. And both of these vaccines activate the immune system very strongly. There are multiple other mRNA vaccines now in clinical trials, including our own, which I'll mention a little bit about now. So our vaccine is called PTX COVID-19B. Just like the others, it is an mRNA vaccine in a lipid nanoparticle. And when we started this process, we chose 3 different potential vaccines to test, and we put those into mice. And the first experiment, we dosed at 20 micrograms each. And we chose the best candidate, and we named that PTX COVID-19B. And it produces the full length S or spike protein, a wild type protein, or in other words, just the same protein that's found in viruses, or at least the original virus. We tested it again at lower doses of 1 and 10 micrograms to be sure that it was a very potent vaccine. And at both of those doses, it did induce strong production of neutralizing antibodies to the S protein. And we did 2 challenge studies, one in a mouse model and one in a hamster model, where we vaccinated the animals first and then gave them the virus intranasally. And we were showed that we were able to have complete protection from lung infection by giving these mouse or these animal models our vaccine before we infected them. We also, because we were going into humans, we had to run what's called a tolerability study at a clinical research organization. So a very high quality lab that is designed to do drug development. And we showed that we had no significant safety issues and high immunogenicity in that model as well, which was just a different strain of mice. And we tried them at 4 and 20. You tend to go higher in the tolerability study, so you can see whether there's any potential problems. And we did not find anything. And I should mention that we also have T cell responses. So when everyone's talking about antibodies, those are B cells in your immune system, and that's definitely very important at catching the virus while it's still in the bloodstream and neutralizing it there. But if it does get into cells, it's very helpful to have a T cell response, so a different immune cell that works differently and recognizes problems with cells. So it's called cell-based immunity as opposed to humoral or blood-based immunity. And this is probably a little too much for this conference, but we do have what's called a Th1 skewed profile. So it's very important. It's what the regulatory agencies are looking for in order to approve testing of a vaccine in humans. Because if you have a different style of response, you can actually lead to problems, toxicities that could affect, could actually make the infection of SARS-CoV-2 worse. And so we have a Th1 response. We used all that data and put together our CTA, or clinical trial application, submitted that to Health Canada, and we received our letter of authorization on December 22nd of last year and started our clinical trial in January. Just to mention, we are currently also working on vaccines for variants of concern, including B1351, which people know familiarly as the South African version, and P1, which people know as the Brazilian version. So here's just a little bit of data. This is from the hamster model. I told you we did a protection study. So we dosed the hamsters first with our vaccine, and then on day 0 here, we challenged them with the live virus, in a very controlled environment, so the virus didn't escape. And what you see is that the mice that were given a negative control, in other words, just a saline injection, not mice, the hamsters, these hamsters lost weight after the challenge, and the hamsters that were vaccinated with our vaccine gained weight, and so showing their health was better. We looked for infectious virus in the lungs of the hamsters, and we could easily find that in the negative control animals that were treated with saline, but we could not detect any infectious virus in the lungs of the hamsters that were given our vaccine. And that's because we had such a high response of neutralizing antibodies shown in the graph at the right. And so this was that's the main method at which it's thought that the vaccines stop the virus is through antibodies in the blood, and we have very high levels of those. And then everyone, of course, is concerned about these variants of concern. And this is an experiment to show that from our mouse study, we could also neutralize the different variants of concern. I show 3 here. So Wuhan is the original virus, and then B117 is what's called the UK variant. P1 is the variant from Brazil, and B1351 is the variant from South Africa. And in this graph, TD tomato is our negative control. It's actually just a unrelated mRNA vaccine that should have no effect on SARS-CoV-2. And what you see is that we did not get any neutralization using that. That's the black dots at the bottom. So there was no neutralization whatsoever of that virus. Whereas if we use the 10 microgram dose, we get very high neutralizing antibodies. And you see that we neutralize the original and the UK extremely strongly. This 10 to the 5, you know, is 100,000. This is a really, that means the amount you can dilute the mouse serum and still neutralize half of the virus. It's a very high titer. And then for the Brazil, you do see a slight decline at the 10 microgram dose, and the same with the South African. But these numbers of 10 to the 5 and even 10 to the 3, you know, represent the what's called titers that would still protect the human against SARS-CoV-2. But now, if we give just a low dose at 1 microgram, we do get, you know, protection of the original virus, the one that the vaccine was designed against. And same with the 117, the UK variant. You know, it's very similar. But if you look at the Brazilian variant, you see that it drops. It basically drops to 0. There's no protection, and the same with the South African. And so that's with what we, you know, a suboptimal dose of our vaccine. And you could also think of it as a suboptimal vaccine. So some, you know, some of the vaccines don't have as good a protection as the Pfizer and the BioNTech. And those vaccines that don't have as good a protection against the original are probably not going to protect against the variants. So it shows that it's important to have a, you know, a really strongly effective vaccine when you measure it against the original, because then you still have, even if you get a reduction of 5 or 10-fold in the neutralization titer, you're still going to protect against that variant strain in, you know, in people in the population. So that's the data about the variants of concern. And then in summary, so there are 2 COVID vaccines that are out there right now, the mRNA vaccines, I should say, and they have been shown so far to be the most effective, and they were the fastest to the market. You know, I'm not trying to say the mRNA vaccines are the only thing. The other vaccines that are out there are still good vaccines, but mRNA did, you know, because the 2 mRNA vaccines were the first to be approved, and they seem to be at this stage the most effective as well. There are more vaccines, as mRNA vaccines, in development, as I said, and some will likely gain approval over the next, probably the next 6 months to a year. And the mRNA vaccines are the easiest to modify and will likely be the first to also be effective against emergent variants. You know, as I said, I think that the variants that are out there, the Brazil and the South African, and also this new Indian variant you might have heard of, are likely to be protected against with the original vaccines, although probably not at a 95%, maybe more in the 80s. But as more variants come out that are even further different, you know, it's going to be a problem, and we'll probably need to do some special vaccines specific for the variants of concern. But mRNA vaccines are also likely to become major players for the prevention of other infectious diseases like Zika, influenza, and malaria, for instance. And there's also going to be therapeutic mRNA vaccines. That's actually what Providence was designed to do. We were a therapeutic mRNA vaccine company, meaning that we produced vaccines for people who already had cancer. So not to prevent cancer, but to treat cancer. And this is another area where mRNA vaccines are going to become more important over the, it's going to take some time still. There's some research that needs to be done, but that'll be coming up as well. And real quick, I didn't want to put it into the commercial, but we did have a press release today where we released our phase 1 clinical study preliminary results. So anybody who's interested can easily just go to our website at providencetherapeutics.com and see our phase 1 results. All right, thank you for your time.
Thank you, Dr. Eric Marcusson. Really fantastic presentation. I appreciate. For the second part here of our event, I would like to invite 2 people. We are going to do a panel discussion. So I'm going to introduce Dr. Claude Pernes, Vice President of Research and Head of the Innovation Program with FIOCRUZ, as well as Dr. Dale Kalina, Infectious Disease Physician, Joseph Brant Hospital, and Associate Clinical Professor of Infectious Disease at McMaster University. Thank you so much for coming. Thank you for being with us today. I would like to ask, our first topic here for this panel discussion would be around compare and contrast of the 2 realities in Brazil and Canada. And I would like to hear from you what you saw in this process from the beginning of the vaccination and how we reach the point right now in terms of challenges, lessons learned, and also perhaps best practice that you have seen that you want to share with us to start this conversation. So I will open to Dr. Kalina to share some of his thoughts on that.
Sure, thank you, Ricardo, and thank you as well for having me today. So to talk initially about what Canada's response has been primarily with regard to vaccines, I think one of the first things that needs to be pointed out is, of course, Canada, aside from newer companies such as Providence Therapeutics, has actually not participated as much as Brazil has, for instance, in a lot of the research and development, nor in a lot of the manufacturing of a lot of the vaccines. So as a result of that, Canada sits at a position where we don't have domestic production of these vaccines, and our entire response to the vaccine rollout has been based on that. So with that in mind, I think that Canada's primary policy has been driven behind procurement of vaccines. I know that we've made international news over the course of the past year or so with the, I'll say, abundance of vaccines that have been procured. But with that in mind as well, a lot of the vaccines that have been rolled out here in Canada, we actually only have 4 that have been approved. So we do have the 2 mRNA vaccines that Dr. Marcusson had been mentioning, the Pfizer-BioNTech and the Mrna Vaccines, in addition to 2 viral vector-based vaccines, the Oxford-AstraZeneca vaccine and also the Johnson & Johnson-Janssen vaccine. So we do have the 4 that have been procured and approved by Health Canada, which is the approvals body for vaccines and medications of Canada. And those are now being rolled out. And I think that at a very high level, there have been a few bumps along the road in terms of the way that the vaccines have actually been procured and also rolled out within the country itself, with the strategy primarily to vaccinate the individuals who are living in long-term care facilities, so nursing homes, and also the individuals, the healthcare workers, to provide them with care. From there, the vaccine rollout was primarily age-based and risk stratified on what level of severe disease risk individuals had, which has been the primary rollout strategy that Canada has had. And only now, over the course of the past 2 weeks or so, have we in Canada been actually receiving higher amounts of vaccine on a daily and a weekly basis, which has sped up the vaccine rollout significantly over the course of the past week. But it does mean that as of today, only around 40–41% of Canadians have had at least 1 dose of the vaccine. And where a 2-dose regimen is required, it's actually only around 3% of the country who's received that, which is far below what we're seeing in a lot of our, especially our G7 counterparts, but a lot of other countries around the world as well.
Very good point. Thank you. Thank you, Dr. Kalina. Dr. Pernes, would you like to share some thoughts on that?
Yeah, so thank you so much for inviting me to participate in this panel. And I should say, well, Brazil is in a similar situation as you just said. Brazil has a nationwide immunization program that started in the 60s, and this was pretty important because it eradicated smallpox and polio, and also because it inaugurated a new phase in the history of public policy in the field of prevention. This program was really able to transform the morbidity and mortality rates radically in the country. And there's 2 public centenary institutions that are part of this process through the production of vaccines. One is FIOCRUZ, the institution I am from, and I must correct that I'm not vice president. I used to be, but I'm not now. I'm just a common senior researcher. So, as I said, FIOCRUZ and Butantan is the two institutions, public institutions that produce vaccines, each around about 7 vaccines against the most common viral diseases. And this is a production that is complementary and not competitive. I think this is pretty important for the country to really strengthen this immunization program. And concerning COVID, the first vaccines available in Brazil were 2 million doses of the Oxford vaccine acquired from the Serum Institute in India. And then 6 million doses of CoronaVac acquired from China. And so far, Butantan has already delivered 46 million doses and FIOCRUZ 30 million. So we have about 10% of the population, the total population that already received the first dose. And it also, the priority was also age-based and risk stratified. But of course, this is not enough because we need, I mean, the country is, continental sized, as Canada is continental size. And the plan is really to make available 100 million doses from AstraZeneca produced at FIOCRUZ until at least this year. And also maybe we will be able to produce the API, and thus the vaccine would be entirely produced in Brazil, not dependent on the importation of the API. And this is the same for Butantan. They estimate another 100 million doses till the end of this year, and also entirely made in Brazil.
Okay, excellent. Thank you, Dr. Permes. I would like to go a little deeper on that, because you see a different participation or different involvement of each country. Dr. Kalina mentioned Canada, that made headlines for their procurement. And Dr. Permes, you explained the two organizations that are producing in Brazil. So let's look at this process of developing the vaccines. We are focusing coronavirus. Can we utilize this technology and this platform to other kinds of virus in the future? And I'm going to enter in variants later, but I would like to hear from you, Dr. Permes.
Oh, absolutely.
Okay.
This platform is the platform we are building will be able to, will allow us to build upon new variants and against new variants, and of course to be utilized for other viral diseases that we face here a lot, such as dengue, Zika, so many arboviruses that are so common here. Yeah, absolutely.
Perfect. And Dr. Kalina, any reason you see, I heard different comments on that, the reasons why Canada bought such a large volume of vaccines. Would you have any thoughts on that for this specific procurement strategy?
I think some of it, just to contrast with Brazil, is I think it needs to be recognized that Canada has about a fifth or a sixth of the population of Brazil. So the number of vaccines that we procured is somewhat in line with what Brazil has procured. But with our small population, that's where I think it becomes somewhat more surprising. Now, the idea, of course, was to hedge our bets on which vaccines would be produced quicker, which vaccines would be as efficacious. And I think we really lucked out here in Canada in the sense that the first 4 vaccines that we did acquire and purchased in large volumes are all very efficacious. Now, at the same time, it does give us the opportunity to allow us to participate quite heavily in COVAX and in the distribution of excess vaccines around the world as well, especially for low and middle income countries, which really does support a lot of the international development work that is performed by Canada in cooperation with a lot of our allies, of course, around the world. But I think that recognizing that we are a country that doesn't produce much of our own vaccine, we don't have a lot of our own vaccine development or production domestically, that really was primarily to hedge our bets and allow us to be able to vaccinate, I think, our population first and foremost, in addition to helping others around the world.
That's fantastic. Thank you. Thank you for this perspective. Okay, the next topic I would like to discuss with you is about the efficacy of those vaccines, especially now that we are facing the variants, and Dr. Marcusson explained very well a few variants and the issues that probably more variants will come. So are we prepared? Those platforms that are we using will be sufficient and will be able to handle variants in the future that are coming? Dr. Permez, I would like to hear from you, your opinion on that.
Okay, so the real world data, shows that the vaccination reduces the risk for hospitalization. And there are some studies showing that vaccination is likely to substantially reduce the virus transmission by reducing the pool of people who become infected. So that's why it's so important to vaccinate as many people we can at the same time, because that would really reduce the chances to spread out the viruses and give the chances to raise new variants. So the threat of the variants we have is really a matter of surveillance. We need to keep monitoring the variants worldwide, and this must be, a huge. It's an effort, a joint effort for all parts of the world. There's an urge to increase the in-country capacity, and in particular in low and middle-income countries, to leverage the existing sequencing resources and to share the results. So at FIOCRUZ, we have groups doing in vitro studies and clinical trials to check if the variants are circulating and if the ones being detected can be neutralized. And as I said, the platform that's being used for the development of vaccine has the capability to be adjusted for the new variants. So I think that monitoring and following up what's happening with variants around the world is not only critical, but it will be really a very good opportunity for us for an effective partnership between Brazil and Canada.
That's a great point. Fantastic. Dr. Kalina, would you like to expand something on this matter?
Yeah, thank you. Just to build on what Dr. Permez has said, looking at what the vaccines that we have available right now and looking at the efficacy, there is data that is coming out all the time right now, both real world and based on studies. There was a recent Qatari study that did look at some of the efficacy of primarily the Pfizer-BioNTech vaccine against the B117 or UK variant and the B1351 or South African variant. And it is just to say that although it works against both of these variants after both doses, but exactly what Dr. Permez has said is increased vigilance and ongoing observation of variants as they appear is going to be important as we move forward, because this is just looking retrospectively, so in the past, about what vaccines we already have and how well they work on variants we've already detected. There will be more variants. There will be other variants of concern that continue to reveal themselves, especially in areas where the virus is not well controlled. And looking at parts around the world, especially India today, but future countries will continue to struggle with this as well, provided we're not able to get vaccines around the world as quickly as possible. And I think it is important to recognize that this pandemic is worldwide in nature. And until the pandemic has resolved everywhere, it hasn't finished anywhere.
Wow. That's an excellent point. Thank you. Thank you, Dr. Kalina. And I would like to enter in an area of future outlook, but I know you guys didn't bring your crystal balls today. So I will ask just best answer for those questions. But first of all, I think it's an important topic is about the research process. And before we know the research process and to develop a vaccine was a long process, years, perhaps, Dr. Kalina, once you mentioned, even sometimes prohibitive. And now we are able to adapt and develop a vaccine with very good efficacy in a short period of time. So what do you guys think is going to happen from now? Are we adopting this new fast-paced platform, or are we going to step back and start reviewing the process and making it longer again? How do you think it's going to evolve from now? Dr. Kalina, if you can answer that first.
I think I'd like to reference what Dr. Marcusson said in his presentation, in that I think one of the silver linings of this pandemic, which, you know, by and large has been awful, but one of the silver linings is that the research process to develop some of these vaccines, especially the ones that are based on mRNA, was a turnaround time of around 8 weeks before they could actually start to get needles in arms for testing processes, which is far faster than what we'd have ever seen before, recognizing that I believe it was the mumps vaccine that was developed in around 4 years. So recognizing that increased speed and capacity of the drug and vaccine development pipeline, I think, or I hope at the very least, will be one of the results of this pandemic, and the capacity for research and development to be completed around the world in greater cooperation, I think will be very important for us to develop vaccines to Zika virus or to malaria, for instance, based on some of these platforms. And the speed will allow more people to benefit on a quicker basis, I think, from these vaccines and from these therapeutics.
Great point. Thank you. Dr. Meirelles, would you like to share some thoughts on that?
As Dr. Kalina said, I mean, this is a new, very new research scenario. And this scenario allowed us, the organizations, to develop pretty fast proof of concept. So as Dr. Marcusson just showed us, it's really amazing what he did in such a short period of time. And this is all over the world. The research process is really different. And I think if we can say there's something good out of these pandemics, it's really it opened doors, or I would say like portals for amazing opportunities for collaboration, for sharing. And there's an additional thing that I think it's very different now, which is the huge improvement in the area of communication. People now can really understand. So the communication between scientists and policymakers and society. So the way you communicate, because these are different languages, right? So the idiom is very different. So the way you communicate and you explain to society how the research process is, what is a clinical trial, how does a vaccine work, what is adverse effect, what, you know, all these terms that we use and the way we communicate is really very different. And it changed the overall research process in a way. And that made a movement towards, you know, people are now willing to contribute to science. Not only financially, but, you know, they are really interested in participation and asking questions. How can we do that? What's the good question? What is a good question? What is a bad question? So at the level of donations and funding and investments has been really amazing. And I should say that this is, for Brazil, this is absolutely new, because this is not the cultural the way we fund science. The usual way we fund here is really depending on public money. And science is performed within the universities and public research institutes. So I think this is really an amazing opportunity, if we can say something good.
Fantastic. Thank you, Dr. Miss. Last question here for our panel. And this is really your opinion, because it's difficult to say what the future is in our lives. But many people are thinking, are we from now on having 2 doses every year? There will be a moment that we don't have to use masks and keep the social distance. How do you think this transition is going to happen? Is something that we can see real changes by next year? What are your thoughts on the transition to the normal life that we had before? Dr. Kalina, you have any comments on that?
So I think we in Canada and in Brazil are in the position that we're able to watch some other countries that are a little ahead of us in terms of what the vaccine rollout has been like and what a bit of a return to normalcy is starting to look like. Of course, in Canada, we're watching the United States quite closely, and also the United Kingdom. And part of what we're seeing is going to be a resolution of, I think, a pandemic that leads to more social activities that you don't require masks, provided you've had both of your doses of the vaccine, and that sort of a situation, while at the same time recognizing that we do need a way of kind of universal documentation for vaccines and vaccine development for things like travel or schooling. And that's the sort of thing that it wouldn't surprise me to see in the near future. I know that the European Union is already working on several of these standards to be able to allow for things that I know I miss, things like travel. But at the same time, perhaps because of this pandemic, especially within North America, we've normalized a lot of mask wearing, for instance, which really does improve the safety for a variety of infectious diseases. I mean, I haven't had a cold in the past year and a half, which is fantastic. But at the same time, while this practice is commonplace in several places around the world, it isn't yet in North America. So perhaps that is more of an ongoing, maybe cold and flu season practice, while at the same time recognizing that future vaccines against COVID-19 might be required for things like the variants and maybe a booster variant, for instance. But I'm hopeful that it won't be a yearly vaccine, but we'll see.
Fantastic, great points. Dr. Permes, any thoughts on that?
Yeah, I would say that, you know, this pandemic has raised a colossal shift in the mindset in general. Not only in the organization, the structure, and the strategies for the health systems, but also in social behavior and economic day life. So I really don't think that we can say that we are going back to the lives we had before the pandemics. I think it's quite the opposite. It's going to be different. So maybe we should, you know, go for Ctrl-Alt-Del and reset the system and, you know, go towards another way of life. So what would be this life? I'm not so sure, but for sure we should learn for these lessons, no? Because there will be other pandemics. This is not a new one. This is a new one, but this is not the only one. We had the H1N1 pandemics some years ago, and we didn't learn from that one. So the world should be prepared. And Bill Gates said a couple of weeks ago that we should learn the lesson COVID-19 is giving us. And I would add for that science, data sharing, and trust are key factors that could help us preventing so many losses around the world we had since January 2020.
Great points. Thank you so much, Dr. Miss. With that, I think I'm closing the panel, but I would like to invite back Dr. Eric Marcusson to join us, as I have a few questions here from the public, and let's see how we are going to address all this. But one that I think Dr. Marcusson or Dr. Kalina, whoever wants to address, is on May 5th, in the United States, indicate that they would waive intellectual property protection for COVID-19 vaccines. So how do you think that this might affect the industry and the production of vaccines and the pandemic, the distribution? How do you think this new possibility to waive the IP protection can impact?
Do you want to take that, or do you want me to?
I mean, I can start. How about that?
Okay,
And,
sure.
and I'd be happy to hand it to you after, as I think it affects you more directly. You know, there are a lot of ways to help low and middle income countries procure vaccines and help them with vaccine development that are not full waivers of intellectual property. And I think the concern that I have, and recognizing that I'm not an expert in IP laws, but the friends of mine are, and in those conversations that I've had with them, there are a lot of precedents set, say with hepatitis C medications that have been produced, in the ability for low and middle income countries to develop patent agreements to produce medications at a cheaper price. And I think the concern that one might have about the complete waiver of intellectual property rights is that it is somewhat of a deterrent for pharmaceutical companies to put in the capital to actually develop some of these therapeutics. I think there are certain benefits certainly to that, but there are a number of ways to get to those benefits as well that also allow for the regulation of the product at the end as well.
Yeah, I think those are very good points, and I think it's also up to the companies, particularly the major ones, to step up and help out with that. It certainly with mRNA vaccines that are very new and require some complicated processes to make, and those processes are not well distributed. Even if you say the IP is not going to be enforced, it's going to take a long time for manufacturing facilities around the world to be able to make these and make them effectively and reproducibly so that they're all the same. A better way to do it is, as Dr. Kalina said, is to have the companies step up and help to distribute the vaccines worldwide. I mean, that's what COVAX was supposed to be, and Dr. Kalina has already mentioned them and how Canada hopes to contribute to that. And some companies have contributed to that. We're a very small company. We hope to get to the position where we can do that, and we would absolutely do that. We're also hoping to get to the position where we can help to transfer some of the technology to sites around the world, so that down in Brazil, the Brazilians can make their own mRNA vaccines. Not with a total loss of IP, but in a way that will make it much more affordable and much more regional for them. And we're doing the same thing. We basically built up Canada to be able to do this by working with a new company called Northern RNA and working with a established company that has it's a U.S. company but has a Manitoba manufacturing facility. So that we're hoping to be able to make 25 million doses this year, and next year we'll greatly increase that capacity. And as we do, I consider it as a good citizen of the world that Providence will share some of that to other countries. And I think that's really more important than just trying to release all IP, because as Dr. Kalina said, it discourages further research into other areas, and also that it doesn't necessarily mean that someone's going to be able to make these vaccines. Now, with some of the different technologies, there may already be, like FIOCRUZ who can do this with the viral vectors. But it's better to help to distribute the technology and the vaccines, I think.
Perfect. I have one here that I heard about. I want to see thoughts. Is related to COVID-19. Countries in general will only receive foreigners that show they were vaccinated. Why some countries are not accepting the CoronaVac vaccine? So if you are vaccinated with CoronaVac, they don't accept. But if you are with others, yes. Any thoughts on that? Is there a reason for that? No comment.
Maybe I don't know whether Dr. Permez wants to handle it, because I think CoronaVac is used in Brazil.
Yeah.
Yes,
well, I'm going to try to answer without being political.
Yeah, that's a real problem, unfortunately. So I'm sorry, I didn't mean to put you on the spot, but
I think this is much more a political decision than anything else. I mean, if the CoronaVac is approved, there is the science is there demonstrating its effectiveness, so efficacy. So I don't really see this is really unfair and perverse in a way, that you accept foreigners having this or that vaccine. So I can go to Europe because I had the AstraZeneca, but I cannot go to the United States to travel. So this is, I mean, this is very difficult in the world for now. I mean, we should maybe so that's what I said, that the thing it's important to have trust in science, and what science, the evidences we have so far. And so monitoring all the people that were vaccinated, how many of them are really protected, and this is key for us, because otherwise, the world is going to become, this is another kind of war then, it's more, it's really a social isolation. And I think this is not good for anyone.
Yeah, and it's a new situation that is having to be figured out, and I no doubt it'll be an evolving situation. For instance, just either today or yesterday, I get my days confused sometime, there was a new study published out of Indonesia that showed that the CoronaVac vaccine in the real world setting was very efficacious with Indonesian healthcare workers. And so we'll have to see as more data comes in, as Dr. Permez said, a trust in science, more data is usually a good thing when it's high quality data. And as we get more data, I think that these kind of restrictions hopefully will change.
True, and policies that are based on data rather than on political will. I think that's one of the things that we've seen here in Canada, certainly, but around the world are public health policies, policies related to vaccines that really do need to be based on scientific data rather than, as Dr. Permez has rightly said, on political will, because looking specifically at this vaccine, it does work. And we've seen evidence of this in Indonesia, but around the world as well. And to prevent people from entering a country based on what data would be the question I would ask. And so I completely agree.
Great point. Fantastic. Thank you. And I just a point here that is being discussed and I hear all the time is about the side effects that we saw with some of the vaccines, AstraZeneca, the blood clots that happened. So is this something that should cause concerns, or is this more a percentage that is very small and it happens pretty much with all the vaccines? So this issue that we saw happening with some of the vaccines, what do you think? What's your opinion, Dr. Kalina?
So that's been a bit of a hot topic, especially here in Canada over the course of the past day or two, because several of the provinces have actually stopped using the AstraZeneca vaccine. Now, recognizing that the AstraZeneca vaccine and viral vector vaccines have a long history of use, we are seeing some cases of vaccine-induced blood clots. And I think there are a few points to make about this. Number one, to recognize the scale of the problem and the scale of the risk, in that the risk is actually quite low of developing these clots, but at the same time, there is that risk. And if you develop symptoms, headaches, muscle pain, shortness of breath, things like that, you should be presenting to an emergency department because that can be treated. But at the same time, the risks of blood clots from COVID alone far exceed the risks from these vaccines, as do oral contraceptive pills or flying on a plane. And so it is certainly a risk-benefit analysis. What we've said time and time again, when the vaccine was available for first doses here, was that because of the risk of acquiring COVID right now itself, it is still far safer for you to be vaccinated. But with changing guidelines and that sort of thing, we are primarily shifting to mRNA vaccines here in parts of Canada. And hopefully that means that excess supply that we have of other vaccines, we can send to others who will be able to make use of it.
Great points. Any other comments?
I should add that, I think this is a pretty important issue to tackle in the surveillance system, signaling the potential adverse events after immunization. This does not necessarily mean that the events are linked to the vaccination itself, but at least it's a very good practice to investigate them. And it also shows that the surveillance system works and effective controls are in place. So I think this would be important also to consider.
Excellent. Great point, Dr. Miss. Thank you. I think it's that. Thank you, guys. We already passed a little bit, so thank you for your time and thank you for your extra time to stay with us here. I want to give really a thank you to all three presenters and panelists today. Dr. Eric Marcusson, co-founder and chief scientist officer of Providence Therapeutics. Dr. Claude Permes, senior researcher and head of the innovation program with FIOCRUZ. And Dr. Kalina, infectious disease physician with St. Joseph Brant Hospital and associate clinical professor of infectious disease at McMaster University. Thank you so much, really appreciate. And I'm sure everybody who attended today is very happy with what they learned today. Thank you very much.
All right, thank you.
Thanks for having me.
Bye.
Bye.
Bye.